Okay, so getting back to the ribbon worms that we saw yesterday, this was John Norenberg, actually from the Smithsonian, who he was able to identify probably the species, the genus and the species, thanks to the great video that we were able to collect. And he said that it's Linnaeid heteronemerdian is the genus and species. And he was able to tell that because of the white borders of the head. And those are cephalic furrows. This guy is an active hunter or scavenger on other worms, mollusks, and occasional fish. Whatever is slow enough to get caught by his proboscis.
Move complete. Thanks, Bridge.
So Matt seems
they
to be
prey usually
us west now, huh?
Yeah.
Prey are swallowed whole, even if it's several times larger than the diameter of the worm.
You can try pushing out
and
the tether length.
When we took our genetic sample of that worm yesterday, we actually saw some
I can follow you around. How far away
what was probably crustacean shell fragments.
The 10-meter move?
Yeah.
In the gut.
That's done.
So wow, it was actually smaller than we thought it was in the video. Of course, we get these super zooms, and I thought it was probably at least a foot long, and it was more like 5 to 6 inches long when we got it back up, and also it might have been much more stretched out when we were first looking at it. But yeah, they were really interesting.
Head back to the west.
And then there was
yeah, kind of push out that
there was a pinkish one that we saw, which was probably a different species.
Copy that.
If not, we can bring you back and try to push south, see if there's anything there.
The first one was in superfamily Reptantia in order Hoplonemerdia. And that one is actually an active hunter predator as well. And I think we were talking about that these ribbon worms fragment a lot when you touch them or try to pick them up.
There's
but these deep sea, at least these
try to trace that up this slope.
Species that we saw yesterday don't usually fragment easy.
That's where we should go.
So thanks for that, John Norenberg and Martha Nazinski, who forwarded that information along to us, and Amanda Demopoulos, who also found it on Twitter. A good example of social media helping us to identify what we're seeing, even beyond the chat room and
is that move stationary?
Science
or
team
is the
that's
ship
on shore.
stationary?
Yeah.
All right.
These pictures and images make their way.
Try and put a toe in here and see if I can get this map.
Taxonomists and scientists all around the world.
Might get a little closer. And then
Yeah.
Of course you can.
Okay. So, I guess where I'm facing upslope, is it easier for me to, instead of coming down on it, just kind of like nose into it?
Yeah. Yeah, just be careful how much you plow into it. The more you plow, the more stuff you mix up, and the more you throw in front of the screen, possibly.
But the current's blowing into me right now, right?
I'm not sure. I don't have a good idea of the current. I think it's pretty light at the moment, so it might linger.
Gotcha. I guess how much closer should I get?
Because
it is in my light pole right now, correct?
Seems like it, yeah.
They seem to be decreasing in extent as we move up this track line. We're a little bit to probably the north of our intended track line, in between waypoints 3 and 4.
Jeff.
Here comes another salp chain passing by.
Alright, video if you want to come in.
We're still seeing some of these quillworm.
We are just kind of aiming at the map just to see if I can get a steady view.
Maybe another one of these gastropod snails. Oh, no, that's a crab.
Maybe.
Another one of the cancer crab
So I'm still moving.
that we've seen a lot of today.
Bridge is zarving out.
Yeah, so you still have to push down a little.
And it looks like there's something on this.
Yeah, I'd like another move. 10 meters. It's bearing 2 through 5 degrees. Speed decimal 2.
Tubes.
Yeah, that's okay.
Luckily it's blowing away. I think I'm putting a little too much on. You can come in a little bit closer if you want video. I'm not really aiming at anything in particular, just... Go ahead, watch one. Tubes at, I'm sorry, she's actually really low on my. Did you hear that, co-pilot?
Tubes of the quill worms, she said.
3 o'clock, alright, come on out, video. Tubes, 3 o'clock.
See those.
Gotcha.
It might be more like 2 o'clock.
Alright, those right there. Up a little bit, I guess. Okay. Oh, the white thing. Are you talking like 1 o'clock and 12 o'clock up there? Okay. You want to come on in on the white worm on the tube video. And coming down a little.
Wondering if this could be a holothurian here.
Come on over. Come on over.
We collected a small holothurian like this at, I think it was Norfolk Seep several years ago. And they were
If you
much
have
more
more,
abundant.
you can come in. I don't know if you do that.
That's max.
That's max. Watch, Lee, would you like me to get closer to that, or is that...
No, that's great. Thank you.
All right. I'm all set when you are, video.
Okay, coming wide.
Yep. Right. Anything else here, watch lead, that interests you?
Try for it here. It's the ship's done moving, yeah.
Yeah, do you want to hop in and grab it?
Just hand him the
Can you do a quick zoom on the quill worm?
nav, right?
It's 2 above the crab.
Okay. Come on in, video.
There's another object on it. Ah, it's a little anemone.
Okay, so
Celebrity sample.
yeah, you want to...
That's good. We just want to take a peek.
All right, come on out.
Grab it or scoop it.
Okay, so if I'm stationary here, I should be able to put in
Auto heading.
auto heading, enjoy log.
Yep.
And then if I leave it,
Should stay.
Move complete. Thanks, Bridge.
Stay there.
Understood. You do make a heading change. 135I, thanks. All right. What was the subject you wanted?
I think
There's the tube with a bunch of stuff growing on it above the crab.
Oh, that's too far away.
you want to... Oh, okay,
Yeah.
I got you.
You want the one with the white guy on it, or the...
No, with the
No.
anatomy on it.
Okay, I guess just point me in the direction of the one that you think you want, and then I'll get a little bit closer and we can zoom on it.
Probably just so you know, high pack went out for a second.
Copy that.
I'll have it back soon.
Okay. Guess my next question is, how close is too close?
All right. Let's take a look at it and see if it's among the living.
Okay,
And then we'll
come on in,
go
video,
from there.
if you don't mind.
It's called the celebrity sample.
Actually, does any of these look decent to you? Looks like the one behind the one with the anemone has a little feelers coming out.
The one with the white.
Okay.
Yeah, the one.
Come out partial video.
And I think it
And
shows
I'm
the
gonna swing
organism.
to a little bit more. I'm gonna come to the right and see if I can find a little bit more. Come out quite a bit video actually.
Yeah, it's
this
that
one
one.
over there.
Yeah. Get your.
Yeah,
And
if
then-
you can just peek at the left end of the quill worm, I think that's showing that it's alive.
Come on in video on this one with the worm.
Well, actually, this is your navigator. Are these going to be pretty delicate or pretty strong? The tubes themselves.
And come in to the very tip, the upper tip.
Pretty strong. Okay. Max.
Max zoom. All right. Well, actually, does that look like it's alive? And that's our sample. All right. Copy that. Come on out video.
Hop over there.
Yeah. Okay. So again, how close is too close?
Oh. You can get it basically between your skids. If
All
you
right.
really, you know. Basically, I would plant left toe to the left of it, essentially, and then you can always pivot
Gotcha.
to go down and right. So yeah, get the left toe basically on the same contour as it, if that makes sense, or below it.
Yeah. So you want to be facing uphill?
Yeah, or slightly to starboard's fine too, or whatever. Understood. Thank you, Bridge.
Okay. Something like that.
So yeah, come down, way down. Yeah, it's within reach. Keep coming straight down, straight down. Yeah, perfect. And then once you get it settled out in a nice landing spot,
I probably should come down a little bit softer.
that's fine.
Everything's much smaller now.
All right, so you just hold right there. Co-pilot, you have hydraulics up?
Spun up.
All right.
And pilot, if you can get a couple of those tubes in one collection, that'd be great. But one is certainly all we need. Just the more, the better.
1 second. I gotta drift in here.
I
So 75-80% is probably way too much.
All right. Copy that. Let me drift up a little bit. And I'll try and get up.
Yeah,
Oh.
you're fine. Just slow movements.
Yeah. You don't need to make large adjustments at
Yeah.
a time.
I think there's quite a few of these up here, so it's not.
Yeah. Try to just wait for it to clear out.
All right, but you have stereo so you can see your position, but
Yeah.
and you have altitude.
I also can see the scars on the ground.
Yeah. I would maybe try going up the hill a little bit. I saw a number of them up there too.
Okay.
Different subjects. Maybe it'll clear off for you.
Yeah.
You can just kind of gently come back in there.
Okay. So I guess
So now,
I was pushing down too hard on the.
yeah, 70% is generally too hard. Yeah, you want to give like a couple, like 10% ahead, 15% ahead, and like just a smidge down. What's your Z-by set? 26. So your little readout should say like 30 or 35 down. Just match those small amounts so that you don't slide around.
Yeah, so that was a good position though, like right between the toes. That was good. Relative to the sample, that was good. Good position.
All right.
think you had it. I think it just you pushed down a little too hard.
Well, I put it into the joy lock, and then it started moving, and I reacted a little too quick. Right, something like that. And then coming down real slow.
Yeah, I like looking at my minute cams a lot too.
Okay.
Just keep the head on the swivel. Yeah. All right.
These are decently sized. Quill worms, that is.
So do I need more than that, or?
You look good on down. Look
there.
good starboard.
You
look
see the
good.
little
Yeah, that looks fine.
So just
crab
hold there.
traces as track marks.
So I should not move.
You need to make adjustment. You want me to joy lock you?
Yeah. So now I can do auto heading.
It's in already.
Is it? Oh. So I should just be able to let go.
Yep.
Yeah, I think I got a little too aggressive with the
Whoa, that's a nice salp chain.
Okay.
joy lock.
You're spun up.
Can you bring mini Zeus on
Very long
one of the
salp
monitors?
chain.
On the right one, actually. I'll walk it over for you.
Oh, sorry.
Starboard enter.
Yep.
Is that what you wanted?
Looks like you're clear. You ready?
Yes.
Standing.
You're pushing into the mud. Oh, well. This doesn't matter.
Can you see my jog, Joe?
Yep.
Is it opening?
Yeah, it does that. Give a little shake. Yep.
Did it drop?
Yeah.
And that
Thank
is
y'all.
specimen number 5, bio, for those of you annotating.
Good on HT2, bud?
Did you want just one, or did you want to grab another one here too, or?
Well, another one would be fine. I had thought you were going to be doing a scoop, so it would be easier to pick up more than one. But they seem to stay pretty much intact when you grabbed them. So
Yeah, I think it may have, like, where the jaws were, crushed it a little bit, but I think either ends of it should still be
Those
intact.
two are coming out.
okay.
Look,
Yeah, let's take one more since we're here.
Understood. Do you see one in field of view that you want specifically, a clean one or a
Well, the
fuzzy
one in the
or-
lower right, is that too hard to manipulate?
I might be able to get it.
Okay.
Like one lighter in color?
Yeah.
Understood.
you want me to come down on the camera?
Yeah.
Or to the left, there's a cleaner one too. Maybe that would be easier.
Yeah, this one over here would be easier.
Yeah.
Starboard inner.
Yeah, whenever you're ready.
Bring a mini Zeus over.
All right.
Pilot, can you hold just for a second?
Yeah.
Thanks.
Stand by.
I think we'll leave this because of it would have to go in a separate bio box, so that
Okay.
was my bad. I
Yep.
thought it would be able to go in the same bio box.
Nope, that's totally fine.
So we want to keep that freed up in case we need it for later. And
Understood.
okay. So thanks.
Yep.
It was good practice.
All right. You are. I'm done over here.
And actually, when you pull away from this area, it'd be nice to take a quick zoom on the excavation of where the skids were.
What is that?
Because we're looking below the layer
Squid?
there. It's kind of interesting.
This is
Just a quick zoom.
interesting.
Squid
Oh,
doing
yeah.
a somersault.
I can
Oh,
do
the
that.
squid is messing with that tube.
Is he trying to?
Yeah, got stuff underneath it or something.
Oh yeah, no, I get it.
Specimen 5.
That dropped on the ship.
Yeah. That's when it restarted. It made the ship.
Jeez. I feel like I came in here in a bulldozer.
He's still there if you want to just drop one by the on the.
Oh, it's relative.
Well, actually, you said you wanted to come in on where the tracks were. Do you have a preference, either left or right?
The one where the bacterial mat is, just to see what is just below the bacterial mat,
Copy that.
What did I move?
just off of center
Oh no.
We can.
to the right.
Yeah.
It may not be as
Like a measuring tool?
clean cut as I'd hope.
So,
Let's let that fade away a little bit,
Yeah, sure.
and then we can come in on it.
Okay.
Okay, video, if you want to start coming in, we can try and. Get some of this.
So the Nemertian that we collected yesterday
Mm-hmm.
had one of those amphipods in it, stomach.
In the stomach, that's right.
Yeah.
And pilot, maybe tilt up and then pan over to the right a little bit to where it's bacteria on the surface. That's where I'm talking about. You can just see the underlying reduced environment. Actually, it's dropped off the screen, so it'd be to upper, yeah, and now to the left a little more.
Is it not always reduced under the surface?
Not always, no. There's enough burrowing on a lot of areas so that it oxygenates the more than just the surface. Certainly around here we've seen this much more commonly. Okay, that's great. Thanks,
what direction
pilot.
are the additional mats here? You kinda
Come on out, video, when you're done.
Are they going uphill or?
I'm not sure. I don't know that we found another one yet.
Okay.
See any more mats here?
So if you have just joined our
What I saw-
dive today,
Trying to
Upper
remember if it
left,
was-
we have
possibly.
been
Yeah.
traversing up a
Oh, yeah, there we go.
moderately
Thank you, video.
steep slope that we came here because we knew that there was a great possibility of finding some of these methane seeps in the bacterial mounds that occur on the surface of the sediments in those environments of the methane-rich water that is emitted from the earth, from the near surface. And
Yeah, you
so
can come on in if you'd like,
these were first identified
just to get a
by
quick
several
zoom on
people, including or several expeditions that showed the existence of the bubble plumes using multi-beam sonar
And that was Okeanos.
from
Okeanos Explorer that were doing some of those first.
this
Come on
very
down on
ship,
the,
and then it's been
anemones,
followed up
believe.
with several other investigations revealing over 500 seeps on the eastern seaboard when just, in the early 2000s we
interest in
didn't know
these,
any
Leslie?
existed. So it's been a great area
we
of
seen these?
study, and using the tool of the multi-beam sonar has been absolutely
this is
essential
worth a lot.
because the sound is bouncing off the lower density bubbles that arise and form sort of plume-like areas so they can be detected in the water column using multi-beam sonar.
Okay. Try it
The
again.
technology has been around a long time, but the water column data is so data rich and takes up a lot of server space and a lot of time to work through the data that a lot of folks haven't really examined the water column data in detail. And so now we're going back to those data.
And this is what we're looking at here is Cereanthid tube anemones eating a salp. So those dark circles that you see there are part of a salp chain, and they've gotten caught up in the sticky tentacles of this tube worm, a tube anemone. Sorry, reading quill worms and thinking, watching these.
I haven't seen a cluster of these tube anemones.
No,
like
I haven't
this.
either. They're usually one at a time, solitary.
And it looks like there might be a quill worm underneath. I see the tube. I don't know if that's a tube of one or maybe that's just another tube anemone.
Do you have any more zoom video, or is that full?
It's about the size of the quill worm.
That's max.
All right.
Great video.
All right.
And some shimmering here.
Yeah, I was going to say,
okay.
I'm clear when you are.
So the reason that we're seeing multiple of them is because they are anemones, not tube anemones. So thanks for that, Tina and Scott.
One just retracted its tentacles somewhat.
All right, clear when you are, video.
And we have a question from the Viz Lab in Charleston.
Oh, yeah.
And it was Beverly who asked, what do you do with the samples you collect and where do they go? And that's
Go
a
in
very
close and see if we
good
can find
question.
some shimmering
The
water
samples that we
Copy
collect,
that on the bacterial net.
yeah, the upper. Let's try the upper left. It was coming down.
they
more bacteria
are
mats,
processed in multiple
coming
ways.
down,
We take samples
into
for
the
genetics,
anemones.
and then
Copy
the
that.
entire sample itself
Upper left.
goes to the Smithsonian Institution. And
Yeah, anywhere right
those
there in center.
samples,
Feel free to come
after
on in.
a bit of time for them to get cataloged and everything, then researchers can request them and examine them themselves so they can be looked at by experts in different taxonomic fields.
Something like
and
that,
then
just like
also,
pan down it, or
genetic work
Just
could be
looking for
done on them
bubbles,
in the
correct?
future as well.
I was looking for a little
And
bit
then
of shimmering.
maybe Leslie can
a
talk
little
about
bit of
what
a
happens
cold
to the
water seep.
geology samples.
Yeah, we haven't seen a
Okay,
a
going
rock
back
in
up to the
quite a
upper left.
while now,
Yeah.
but we do collect rock samples when we feel that they can represent an area that
would
we
you
are
prefer it to be a
diving on.
slower turn or a
And
faster turn
we,
video? How do you?
No, that's
Photograph
fine.
them,
It's-
we weigh them. We
it's
don't
not
do
really
too much
leaping out
here
at us.
on board. We do remove any organisms that are attached to them, and we save
It may
those.
have
And
been like a
the
shrimp or something that
organisms
got in front of
removed
us that
are
gave
sent
us that
to the
shimmer.
Smithsonian
Yeah.
along with the other biological samples.
Okay.
But
All right, come
rocks
on out when
and
you're
also
ready.
sediment samples, we do make collections using the scoop. We don't have a coring device or a suction device, so we do
See
the
if
best we
can find the
can
next one.
with a scoop. and we send some of that
Is
raw
this still
material,
stationary?
or we put
Yes.
it into
Looking for the
bags,
next,
and I sieve a small
match.
portion
That
of it
one
so I can
dead
see
ahead,
what
maybe?
the larger size
Or
fraction
is that just my
is.
eyes?
Out in the distance.
And all that material
Yeah.
gets sent
Looks
to
like
Oregon
one.
State University,
Yeah.
and they archive and
It looks like the little
analyze,
out there.
particularly the rock samples. I
Wanna
don't know
turn
how
to that
much
one?
they actually
Get it
do
over
with
that
the
way,
sediment
Jeff?
samples,
Yeah, turning.
but they
Do you want me to
slice
look up slope
a
a little
rock
bit
sample,
and see if there's not one up there?
just a fragment
Yeah,
of it, into a very thin sheet and put it on a
Looking at
micro
like 250
a
maybe.
micro slide, a glass slide, and polish
20
that
years good?
thin
Yeah,
piece
it
of
should
the
be fine.
rock down
Starting to push out
to
quite
a
bit,
point
right?
where it is microscopically thin, and
Yeah.
it
Bridge is
transmits
over now.
light through it, and we can look at the
Yeah, I'd like to
mineral
move
content
20
of the
meters,
rocks
bearing
using
250 degrees,
polarized
speed
light,
decimal 2.
and we can identify the different minerals based on its optical properties under polarized
Feel free to
light. So
come
it's
in on
really
that
interesting
video if
and
you'd like.
beautiful
Just to see if we see any shimmer.
microscopic images of and we call those thin sections. So we can understand a little bit about the rock
Could
structure.
you come on in video?
But as I said, we haven't collected a rock in a while, and most of the rocks that we have collected have been really compacted and almost fully consolidated, almost lithified mudstones. So you can imagine taking this sediment, not the white sediment, but the brown sediments, and burying
Taking
it,
it out,
squeezing out the water, and those grains start to bind together with some of the mineral matter in the water, precipitating out and binding it together, and they form layers of rocks in the geologic
All right,
record.
go ahead and come out. I guess we'll try and find another one.
So we get a lot more biologic samples on these dives, though, and a lot more range of organisms than we have, and range of sediment types or rock types.
And it's a very careful process selecting what we do want to collect. So some of the criteria that we use is are whether it's a species that was seen often at the site, so to help characterize the site, to make sure we have a good identification of the main species that we observed.
Something white
The
up there.
next is if it looks like something either new to science or a range extension of something that is known. We will make a collection so that we can verify that to make sure it is correctly
Sorry, say that again.
categorized. And we get a lot of input from the other scientists
Copy that,
in
coming
our
back.
chat room and who call in.
It'll be right as center.
Right, upside
So all of these factors will
back.
play into whether we make a collection or not. And we have
Should be coming
4
in center.
bins on the ROV, so we can make a maximum of 4 major collections, biology
There he
collections
is,
per
just
dive.
to the right of center.
Okay. Get a little forward and come on in.
We're gonna.
So I think it's worth adding that one thing that's pretty unique about this
Right,
thing,
like
I know
at the
it's
head of that
science
snipe eel, or?
usually what work. All these scientists who are on the call, most of
See
us
the
go
little head of
to
the fish
SEEP, which
sticking
is
out of
one
it?
or two other scientists and the graduate
Yeah,
students,
come on
et cetera, to do our
I
own work
gotcha.
often. But what's really unique about this is that because we have all these scientists out here,
Coming
plus
down.
NOAA has taken the perspective that the science
Take
should be
more if you
available
got
to
it.
everybody. None of the samples are not necessarily being collected for one person. They're available to really anyone
This is what you
in the
want to watch,
community
Lee?
who has a scientific interest in doing something
The
with them.
dust storm.
Yeah, that's so true, Carolyn.
Click remove.
This is a very unique situation, and it's
Yeah, you can try.
I think it's so great for science to have all of this input as to whether what you're seeing is unique. If you go out
Right,
as
come on out,
a
video when you're
smaller
ready.
scientific group, you have just the knowledge that's on the ship at one time. But with this telepresence,
I just
we
need
have
to fix my comms.
just extended knowledge to all types of different scientists, and it
And
really
then,
makes all of the information come together so
push out
quickly.
once you do that.
Copy that. Oh, yeah. Sorry, I could hear OKX pretty good, but I
As
couldn't hear
we've
the
said
watch
many
lead.
times, it's almost impossible to be an expert in everything you'd see out here. It's all the different types of biology, the different groups of organisms that you see, as well as the oceanography and the geology.
Let me see.
So having input from different types of scientists really makes you be able to characterize an environment much more comprehensively and quickly too.
Move complete. Thanks, Bridge.
So thank you for the original question from the Viz Lab at the College of Charleston, and actually it was from Kimberly.
Yeah, you can,
we were
change your heading if
told
you'd like.
that the name was incorrect to begin with.
140I.
So thanks, Kimberly. And yes, I do love rocks and sediment.
Yeah, another
Ask
one.
any of my students. So we just took a peek at a fish in one of the burrows, and
There
that's
we
the
go.
first time that we've seen a fish in a burrow that was not the same. That was actually one of the
What is
southern
that?
hakes. And
Yeah,
every
right.
other time we've seen a fish, it has been...
With the large.
What's the type
those
of
down.
rye fish?
Rye mouth.
Rye
I think
mouth.
is what they're.
Sorry, I should write that down.
See the left half is
Keep getting it wrong every
body
time.
and
Is
then
that-
it's buried
is
a
that
little bit.
Pretty
the
big though,
white
isn't it?
things.
Yeah.
What
That's
is
funny.
that?
It
Coming
looks like
on in
a
video?
buried shrimp or something, but it has a very long...
I'm going to come back a little bit.
That's very unusual behavior.
There's not much motion.
Tuck itself in. It's taking a nap.
Come in a little bit more if you'd like.
With a tiny little pout eel.
Oh my goodness!
Checking
Look how
it out.
tiny that guy is. And an incoming salp. Well, that sure is interesting.
Tiny little gastropod that just went
Push
off the
forward
lower
rather than
right
rocking
of the
the
picture, too.
And there's a
hammer too
little
much.
Copy that.
quill worm, it looks like,
Look
in
at
the
that
upper
little
left.
gastropod.
Wow, that is tiny.
There you
Goodness.
go.
See,
I'm just
how many,
always amazed by
guys
this
on your light bar?
zoom
Isn't that nuts?
and
On
the
your aft poles?
lovely
Oh, yeah.
images we get.
Those krill or
So
shrimp
he is
or-
alive. He's
It was
moving
krill
around.
earlier. Not sure what it is now.
Alright, good when you are video.
Huh. Interesting to see them buried.
It moves complete.
And
Copy that.
he looks as though he has burrowed himself. Not that, because it's undisturbed around him except for right where his head is.
Alright, let's move along to this other mat, I suppose.
And Pilot, can we turn on the lasers, please? We've
yeah.
had a request
Go
for
ahead.
that. Thanks. Not just for this guy, but as we're in transit. It's easy to forget how far we are from the
It's a
substrate.
pretty big shrimp.
That is a big shrimp.
It is a big shrimp. And I don't think we've seen any shrimp on our expedition that bury like this.
If you want to just do a partial zoom, or snap zoom, is that what it's called? Like not all the way in, but just enough to see if there's anything there.
Wow,
is right.
it's a
Partial.
quillworm convention.
Okay, and then, so are we looking for bubbles? Is that the idea of the mats? So if we don't see bubbles, we should just keep moving to the next one, or what's the thought process here?
Are you asking me or watching?
I'm asking anyone that's has an answer.
Actually, if you're asking whether we should look at each of the bacterial mats, right?
Yes.
Yeah, I think if we could get a good zoom on each one that we come to, and we'll be trying to see if there are bubbles or not, and anything else that's might be living in the mat. So that would be wonderful.
Copy
Thank you.
that. Feel free to come in video. I know we're a little far away, but I'll move closer as we come in.
Yeah, well, there wasn't much in this one.
Yeah, it looked pretty small. Oop.
We're looking for the jackpot with mussels, lots of mollusk.
Copy that. Alright, you can go ahead and come out and we'll see if we can find a bigger mat. Looks like there's one ahead. Go ahead for a snap zoom.
It's interesting, we're still seeing these little patches of the bacterial mats where before, when we were really close to our strongest target, we saw the most extensive coverage of these mats.
Nothing.
Oh, look at this worm. You can see how it moves.
Wow.
Come on.
That's interesting.
He's got a mouthful of the sediment, it looks like.
Yeah.
Feel free to take as much or as little zoom as you want, video. I'll try and follow this guy on his commute.
It's a serious core workout.
See, looks like an inchworm trying to drag something.
I wonder what just went buried. Oh, I guess that's another one of these worms that's
Yeah.
buried over there.
Coming out now. Huh.
He might have a hard time dragging his house out of the mud.
Is this how you're supposed to dance the worm? I feel like I've been doing it wrong my whole life.
Can't say I've ever done it.
Can we get a little tighter zoom on that? I
Yeah,
would
video
love
if you
to see
can. I
his-
can. Do you want the bottom or the top?
Of the head
Yeah,
of
that
the
one's-
top,
Well,
the one that's moving.
Gotcha.
I want
It's
to
got
see the
spikes on it.
near the mouth what is grabbing onto the sediment.
Oh, I wonder if he's going to eat whatever this little red thing is.
Looks like it.
Oh, yeah. Oh, he takes it inside.
He doesn't want to
shy
be
eater.
filmed. He knows we zoom in on the kill usually. No privacy while dining around here. Well, I guess the show's over.
All right. Feel free to come out when you're ready.
Thanks.
Video.
Oh, that was great.
Is
That
that
was.
just the head of a worm? There?
Yeah. So the worm has the rest of its body in that tube.
Well, I mean, this one
Yeah,
right
but
here.
it looks
it
like
looks
the
like
worm
it's-
head over here.
Oh.
Severed
That's
worm
one that
head.
maybe.
Or is he just underneath?
Hard to say what that is.
Yeah,
No,
I
I
bet
think
his
that's
tube-
poking out.
Yeah,
Yeah.
his tube is in the sediment
Yeah,
there,
you
the
can
rest
see
of
the
it.
little tentacle features.
All right.
The amazing
I
world
guess-
of worms.
Go on to the next mat. And we're coming up on an hour if navigator or co-pilot wants to hop in.
Scott France
Looks like
writes,
there's another one up
obviously
there.
having such an unwieldy tube that has to be lugged around is a benefit. It's likely for protection, and I guess so.
Yeah, otherwise that's an awful lot of energy to expend.
And privacy.
Oh, yeah, and
Playing
privacy
connect the dots.
for those shy worms.
The bacteria mats be the dots.
This looks like a little bit more of a... If you want to take a snap zoom video, see if we got any bubbles.
Didn't see anything, but
No.
Not quite as active.
We have two more of those mollusks at the top that we've been seeing
What do you-
yesterday and today. And
Like a zoom on that?
yeah, and then the thing in the upper, oh, that's just an eel pout up there. Yeah, that would be great. Thanks. And we've made one collection of these today.
Feel free to take as much, or is that full zoom?
yeah.
All right. Would you like a closer view of this watch lead, or?
Oh, no, that's great. Thanks.
Okay. Feel free to come out when you want video. I don't see any bubbles.
If we can just look right, just get a nice scan of it, that would be lovely.
Okay, coming back to starboard. Oh, jeez. Need to wash my toes.
There's a few more of the gastropods on the right side of the field of view now.
Yep.
we have
Yeah,
interest in
looking
going
at
to that upper right.
screen 2 or camera
dead
2, you see that there's not nearly as much in the water as there was earlier. We could hardly even see D2 from Sirius's perspective when we first landed, and in the
Trying to figure
first
where the next one is, Jeff.
major,
I don't
2
see
hours
any
of the
pictures.
dive, we saw just a
Alright,
lot
feel
of
free to come
biota
out
in the
video.
water.
Thanks.
So we've risen above that layer, apparently.
Yeah, and now there's a lot of sedimentation in the water, though. I mean, it's still a very
Full out.
blurry view of
Trying
D2
to find
from
Oh,
the
there's another
surface.
worm walking uphill.
And we are currently at a depth of 435 meters, and we began at
We need to be
519.
making our way uphill, right? Or trying to.
A little bit, not. Try to find the.
Just chasing biomats,
Yeah.
wherever they happen to be.
We've been taking water samples every waypoint,
Okay.
but we kind of.
And there were several potential
there's a
seep
map there
sites
uphill.
Yep.
noted along this track that we've chosen. So we're hoping to see
There we
more
go. I was
and
going to say, we haven't looked
patches
up
throughout the
that
dive.
slope in a while.
Looks like we
Certainly
have one
been
after
rewarded
these two
so
as well.
far.
further
As we
up.
landed, we saw a field of these
Let's
bacterial
go to the bigger
mat
one first.
areas.
Well, actually, this is your navigator. This is about as close to waypoint 4 as we're going to get, and we're pretty much between waypoint 3 and 5. Would you like us to take a water sample here? Understood. All right, pilots. We can take a water sample, co-pilot.
Come on in on this.
We'll drop
I'll
the
swing around.
Done.
Still don't see any shrimmerin.
No?
It's like spec 06. Watt.
There. Come on out if you're satisfied. And I guess we'll
I don't know.
continue up the hill. Oh, there's another buried shrimp. Slightly uncovered.
Maybe 225 nav, we can do another 20 meters.
225?
Come on in on this video.
5, affirmative.
The shrimp that's buried.
And here's another of those partially buried
Bridge
shrimp.
is RV nav.
Curious.
Yeah, like a move 20 meters, bearing 225 degrees, speed decimal 2.
I see what you mean by going
Good
forward
copy.
and backwards rather than tilting the camera.
Yeah.
Just a little bit smoother. Okay. I
That
think I
was the
saw.
fourth Niskin, is that correct?
4, yes.
So we have this one straight, and we also have one up to the starboard, or the port, I believe. Have we seen the rat tail today?
Ask about
Sorry, but has the water sample been collected?
Yes, it
Okay.
Yeah, we just took
Great,
it
okay.
2 minutes ago, yeah.
Thanks.
We got something here buried as well. Almost center. Oop, that's the wrong way. I think that's. You
Go
see
ahead,
that right
Lexi.
above the lasers video?
Oh, understood. Yeah, we got it.
So yeah, another type of grumpy fish.
And this is our at least third batfish of the day. I don't know if you saw one when I was at lunch, but I definitely like them.
Here we go.
Uh-oh,
So
we
you
might
gonna eat
see
it?
feeding behavior.
Go for it.
Maybe.
Oh,
Come on.
Must not be
now.
hungry.
We've
They
already
have
seen quite a few of
modified
these
fins
today.
so they can walk along with sediment. But they are little angler fish. So they are looking for
Pilot
food
and- or
above
co-pilot
them,
and
not
nav.
in the sediments
If one of you wants to
below.
hop in, just let me know.
Understood.
Pilot.
Pilot, you got
So
2
we
minutes
seem
to
to
show
have
me
been
something amazing.
wandering
All right, you
away
just
from
missed the
some of
last
the
hour
seep
of-
areas.
No,
Interesting things.
2 minutes.
you don't understand. You got 2 minutes.
Can we scan over to
show
the
me something
left to
amazing.
see if there are patches that we're missing?
Copy that.
The bacterial mats.
Video, you have an open
Black-belly
mic.
rosefish in the distance.
That was intentional, Pilot.
I think it's still open. It was,
There's something shiny up there.
We're starting to see some mounds that we haven't seen today, but we have seen in the past.
Right-
So some
right in the
organism
mud.
is generating these little mounds.
That looks like another southern hake here that's buried.
Coming back.
Oh, yeah. Missed him. Right there? Or is it a crab claw? No.
It's definitely a fish.
Yep.
Sometimes it looks like, I know once we...
Feel free to come in, video.
Oh, I see. His whole body is lying down.
Coming
Yep.
back.
And that eel pout is curling around him.
I love how they just make a little blanket over them. A blanket of sediment.
All right, feel free to come out.
A mud bath, Amanda.
Mud bath.
Going to the spa for a day.
I haven't seen a mat recently.
All right, video swapping out.
thanks, Roland.
Thank you, video.
Understood. Going to 5 degrees to starboard. Thanks, Bridge.
Slurped out of the sand.
Delta's pretty low. Okay. It's lower than I run it, but understood.
Try to push out a little. Oh, there's a mat.
Looks like with the slope there.
If you want to do a snap zoom here, video, just to see if it's bubbling. Nothing. Alright, come on out. Let's see if we can find another one. I see one down to the right. I mean, we still want to make our way uphill though, right? If we can. Alright, let me just look up and see if we can. There's one there.
There you go. I think it's zone. I see. Yeah, there's two of them there. All both the same direction.
Have we seen this fishtail before?
Yeah, this is a hagfish. We've seen quite a few of them, and this one looks like it's burrowing its way into the sediment.
Oh, there's a whole bunch up there.
Watching it.
Yeah, I see a trail.
Yeah.
Of bacterial mats bearing uphill. Looks like
Great.
a little bit west of south, which is
Yeah, we were just going to suggest moving up the slope a little bit. That's great.
Copy that.
We'll let these bacterial mats guide us to the larger areas.
Is that a crab or?
Around the edges.
I have bridge.
I think that's one of those snails.
right now.
That's a big snail.
That shouldn't be possible.
Or is that a? There's a big mat. Quick zoom on this little one here. and then if nothing, then we'll move our way up to the big one.
Wonder if that's a burrow that an organism made, or if that's from actual seepage, that
Yeah,
larger
it's hard
hole.
to tell.
Okay. Come on out.
There are other holes like that around that don't have bacterial mats associated with them. These quillworms are really funny.
Yes. Quick one here, and then if not, we can go up to
Walk
the
across
upper
the
left.
sediments everywhere. They're quite active.
Yeah, I don't see any shimmer. All right, feel free to come out, video. And there should be that guy. Ooh. Too much. Go ahead and zoom in on this video as we get closer.
Worm horror stories are coming in now on the chat room. Fodder for nightmares.
Oh,
I guess in that situation,
or for sci-fi, sci-fi films.
Approach too fast? Is that why that was? All right.
Apparently there is a king worm that can be 2.5 meters long.
Oh my goodness.
That's something like close to 8 feet. Oh my gosh.
I really hope you don't see one of those.
We will not be collecting
so here
that, I guarantee.
I'm floating away, so I need to push up. All right.
Did I see that was from Australia?
I didn't read that far back, I don't think.
Is there another one of
This
those
pile of
fish?
quillworms.
Yep.
Yeah, another southern hake buried on the left in a pile of quillworms.
I keep pulling towards me to...
You can just pivot off that tugboat.
Yeah, can we do a quick zoom on the quill worms, please?
Right in front of me?
The whole pile of
Yeah.
them.
Yeah.
Come on in, video.
Let's see if we can figure out what they're eating. There's a gastropod, but we're not sure who's eating whom.
Anyone
2
else?
gastropods. Look at 3.
Oh,
4.
wow.
At least 4.
I guess there's
My
some
gosh.
white, fleshy.
Yeah, I think
Probably
that
some
might
kind
be
of
the
tissue.
gastropod they were working on.
Do you have any more zoom video, or is that it?
That's it.
Or is that an amphipod? No, I don't know.
Oh my
It
gosh.
looks like the 2 worms are playing tug of war on the piece of tissue that they have there.
So, in that case, I'm going too close.
This would be a good scoop sample if you wanted a quillworm sample. Wow. So it's interesting that so many of the quillworms have
Something like that.
sediment still attached to them, and so many are clean. Oh, look at that one coming out. Oh,
Oh, you can see it's kind of biting.
yeah.